So like I said, I'm not going to ask you to redraw this, but when we think about a protein, we know that it's made out of a few elements. I'll remind you: carbon, hydrogen, oxygen, nitrogen, sulfur a lot of the time. The primary structure down here is where we have an amino acid chain, where I took a few amino acids, or sometimes even 100 amino acids, and linked them together in a chain. We know how that chain gets made. It gets made because of peptide bonds. So far, that's all that's happened. I've put them together, they're in a long chain. But in reality, that's not the last thing that happens with proteins. More things happen. In the secondary structure, I've taken my chain and I've given it a twist. I'm being very generic here. There are specific ways that this happens, but the chemistry to explain that is beyond the scope of our course. The idea is that instead of one straight line, now it starts twisting and turning over itself. Why does this happen? There's a thing in chemistry called a hydrogen bond. Its definition is part of chemistry, not biology. But the quick definition is there is attraction. If you remember when we talked about water, water is a polar molecule. That means one side of it is positive, the other side is negative. Amino acids have that as well. That means that one side of one amino acid and the other side of another amino acid are kind of attracted to each other. They're not making an actual chemical bond, but there's an attractive force there between them, and that's what makes it kind of twist up. The tertiary structure is where we start getting a little bit more 3D. So instead of just a chain with a twist, now I'm making sort of a ball-like structure. And in the quaternary structure, which not every protein has, you'll notice it's like I've got 4 pieces that I put together. So instead of just one 3D piece, now I have a few 3D pieces that I put together. What I want to get at here is that the primary structure is based on peptide bonds. That's an actual chemical bond. It's quite hard to break. The rest of this is all based on attractive forces, things that are causing it to twist and bend and roll into a little ball. Those are easier to break. Now, does anyone want to ask any questions about that? I won't ask you to draw this. I just wanted to give you a visual of what's happening. So then we'll talk about a couple of things that can happen to proteins that will affect, in particular, this side of the picture. Like I said, a peptide bond is really strong. It would take a lot of energy, and it would be hard to break a peptide bond, but these attractive forces, that's a little bit easier. So there are 2 alterations to protein structure that I need for you to be aware of. One of them is called denaturation. So whenever you see ation at the end of the word, that tells you that there's some sort of process happening. So I am unnaturing this particular structure. I'm taking it out of its natural state. Denaturation is when proteins are, and let's make sure we can read that word because it's a big deal, temporarily changed by physical or chemical processes. So when I denature a protein, what am I going to change? I'm going to change the secondary or the tertiary or the quaternary structure. Not the primary structure, that's the peptide bonds, those are hard to break. But any of the parts of their structure that cause them to twist or make a little ball or group together as more than one subunit. There's a picture here. The idea in the picture is that the normal protein is all twisted up, and the denatured protein, now it's like I've straightened it out. Now I know that this is starting to be a really archaic reference. But I'm going to talk about a cassette tape. Have you all seen one, heard of one, seen it in a museum maybe? I used to have to watch movies on VHS tapes and listen to music on cassette tapes. And every once in a while, your stupid little brother would take your stuff and wreck it, and the ribbon would come out of the tape. And that's my analogy for a denatured protein. My ruined tape. So what did you do when your stupid little brother ruins your stuff? Well, you take a pencil, you stick it in the little groove, and you wind the ribbon back up. And you hope that it's not ripped or cracked and that it still plays your music. My normal protein, I can get it back by rewinding the tape. So putting a pencil in and getting all the ribbon back up into the cassette so that I can listen to it or watch it again. So denaturation is temporary. I can get my protein back. I can rewind it up. I can make it look its normal, natural state again. So what are these physical or chemical processes? It's usually temperature or pH. Those are the two most common ones because those are the conditions that proteins encounter in your body. Now you'll notice I used the word enzymes here. Remember, enzyme is a function of proteins. And since enzymes are the ones that we're going to talk about the most, I'm starting to insert the word enzymes here a whole bunch. But if it gets a little bit too hot or a little bit too cold, or the pH gets a little bit too high or a little bit too low, if there is a surge in electricity, or if a heavy metal is around, a heavy metal is literally what its name sounds like. It's a metal with a large molar mass. Heavy metals can have toxic effects because of this, because they can denature proteins. Key idea, change is reversible. I can get my protein back. So denaturation can happen, but notice down here, renaturation can happen too. I can put it back. So what does this mean? This means, for example, if a person has a fever, they're too hot. Some of their proteins might not function properly while they have the fever. When the fever goes away, the proteins will go back to their normal function. Same thing would happen with hypothermia. A person's too cold. Well, as long as they warm back up, their proteins might go back to the function that they had. Now the second thing is coagulation. And you might have heard this word before when talking about blood. Coagulation is something you might see when your blood starts hardening and forming a clot. That's what the word coagulation is used for. What does it mean? It's when proteins are permanently changed by the same processes listed above. So it could be heat, it could be pH, electricity, heavy metals, but the idea here is that it's permanent. It's not going back. What will have happened here is that the 3D structure is completely ruined. And then the small fragments of proteins will reattach to each other in a different way. And my example here is cooking. Specifically, that's an egg because we know that eggs are made out of protein, but the same would be true of cooking anything that's made of protein. You can't uncook a steak either. But when you cook something, that's not mild temperature. That's usually fairly excessive temperature, way above normal temperatures that you would find in a living organism. And that's too much. The protein can't handle it, and the 3D structure, the chain with a twist, the structure will be ruined so much that it can't get back to the way that it was. So coagulation is permanent, denaturation is temporary. They're caused by the same types of things, but one of them is the end of the line for the protein. Does anyone have any questions about that? Now I want you to notice something specific about these conditions. Notice that I said it could be too hot or too cold. pH could be too high or too low, and that I didn't give a specific number. And that's because not every protein functions best at the same temperature or at the same pH. And so I've left this general on purpose because it's not the same number for every single protein. Does anyone have questions here that they want to ask? So we are going to focus on enzymes. It is one of the functions of proteins. We will focus on some of the other functions of proteins as we go through the course, but for right now, we're going to focus on enzymes. Enzymes almost always end with ASE. Not to be confused with carbs that often end with OSE, so this is slightly different. But this is a good way to recognize that we're talking about an enzyme. So what is an enzyme? It's a catalyst. That's a chemistry word. And I've defined it here in case you don't know what a catalyst is. If you do know what a catalyst is, be picky when you're writing stuff down. You don't have to copy every word that I've written. Perhaps you say, I know what a catalyst is, and I don't have to write this. What is a catalyst? It speeds up chemical reactions, but it is not altered. And because of that, it can be used over and over and over and over and over and over again. So enzymes are proteins that help chemical reactions happen faster, but they're not actually getting changed. They're just speeding it up so I can use them again and again and again and again. Now the second point is really important because it's something that I would ask on a test, and it's something that I've seen, for example, on diploma exams, because you keep talking about enzymes in biology 30 and you talk about enzymes for sure in chemistry 30. So if you're going to take either of those courses, this is an important point. They only accelerate the rates of chemical reactions that already occur. They're not going to make something happen if it wasn't already going to happen. They're not magic. They just help things that are already happening happen faster. So I'll give you an analogy of like what a catalyst is. Remember, catalysts are speeding things up. They're not getting changed by what's happening. They can be used over and over again, but they don't make things happen that weren't already happening. A catalyst is like your teacher during scheduled help. I can help speed up the process of your homework getting completed. I'm not used up in the process. Many people can ask me questions and I can answer it over and over. I could even answer the same question over and over again. But I cannot make something happen that wasn't going to happen. So if you aren't here for scheduled help, well, I can't make you do your homework from a distance. If I had that kind of mind power, I would be somewhere else right now using my mind power to do awesome things. But I can't make it happen if it wasn't already happening. So the reaction has to be happening already. The catalyst just makes it happen faster. Now, does anyone have any questions about what a catalyst is? Tomorrow we're going to do an awesome lab. It's more of an activity than a lab because it's really hard to do enzyme labs because you can't see them. But you are going to be the enzyme in this activity. It's going to be fantastic. So I'm going to show you a little bit about what it is that enzymes are actually doing. The model that we typically talk about is something called the lock and key model. In this model, the enzyme will be the key and the substrate is the lock. I even like to use the word door because I think it gives you a better visual of what's happening here. Now the picture that's on the left here is very similar to the picture that's on page 215 of your textbook. So if you would prefer to look at that picture rather than redraw things that are up here, that's where you can find it. So first thing we'll do is talk about this word, substrate. We know that an enzyme is a protein. What is a substrate? The substrate is the substance that is reacting. In my particular example here, and in the example in your textbook, the substrate is sucrose. So whatever molecule, for us we'll typically talk about macromolecules, whatever molecule is going to do a chemical reaction, that is the substrate. Now I want you to notice something here. The substrate is sucrose. The enzyme, very conveniently, is sucrase. Lots of enzymes are named like that. They sound exactly like the thing that they're helping, but the ending of the word is changed. Not 100%, but a lot of the time. So there's an enzyme called sucrase. There's an enzyme called maltase for maltose. There's an enzyme called lactase for lactose. Now, a really key thing is this. An enzyme must come into contact. An enzyme can't do its job if it cannot physically touch the substrate molecule. So you might have learned, it's in grade 9 science, I used to do a super awesome lab about this, that if you crush something up or break it up into tiny pieces, the reaction happens faster or more vigorously. And this sometimes is why, if more enzyme can touch more substrate, it should happen faster. So I'm just going to add the idea of surface area here. The more surface area there is, the faster enzymes and substrates can come in contact with each other. So in my picture, my enzyme is a big purple blob. It is that way in your textbook too, but the blob is shaped slightly differently. Sucrose is my substrate. They're both just sitting around. Now they're going to come into contact with each other. Once they're together, so I'm going to make my circle half green and half red. Once they're together, now I call it the enzyme substrate complex. Now, how did they go together? Well, if you notice on my enzyme, there's a little spot, like a little hole, where it looks like sucrose would fit perfectly. It would fit exactly into that shape. This little spot, I'll make it blue so I can color code it, is called the active site. So the active site is a match for the shape of the substrate. Now, is it an exact perfect match? Sometimes it's not perfect. Sometimes it can wiggle a little bit to fit the way it needs to fit. But the active site needs to approximate the shape of the substrate. Now, what can happen once they're attached to each other? It could be catabolism, or it could be anabolism. Remember, catabolism is when we're breaking it apart. Anabolism is when we're building it. A lot of the pictures that we use and that your textbook uses to represent enzymes show catabolism. That's what a lot of the pictures show, because sometimes it's an easier drawing to make, but I want you to know that either thing can happen. In this particular picture, I can see that it's catabolism because my sucrose is getting broken in half. And how did it get broken in half? Hydrolysis. There's a little molecule of water. So what did my enzyme actually do? An enzyme is kind of like a matchmaker in this sense. It helped sucrose and water find each other. And then sucrose and water could do a hydrolysis reaction. It didn't get changed. Nothing happened to the enzyme. It just was a spot. It was like a meeting point or a way for them to get together faster. Here are the products. So I can see, since I started off with one large molecule and I have two small ones, that definitely catabolism is what was happening. So I've told you what enzymes do and kind of what it looks like, but I didn't tell you how. And that's a key part of this equation. How do enzymes work? So I know they're making a reaction happen faster, but what are they actually doing? The thing that they are doing is they are lowering the activation energy. All chemical reactions, even if they're going to produce energy in the long run, require a bit of energy to get started. There has to be some input. Every chemical reaction is like that. What does an enzyme or a catalyst do? They lower that energy. That means that to get started, less energy has to be put in. So it's easier for the reaction to get started, which makes it happen faster. The reason that I need this energy is this. Anytime a chemical reaction happens, I have to break some bonds and I have to make some bonds. That's what's going on. So I'm going to rearrange my molecules, and energy is required to get that process started. So in the picture here, I'm illustrating breaking bonds, but the same could be said for making bonds. I have an enzyme, and I have a substrate. Notice how the shape of the enzyme and the shape of the substrate match each other. And I picked this picture because it looks a little bit different. The other ones are all geometric shapes, and this one is showing you that as long as the shape matches, they can fit. The active site, keep in mind, is specific. If you don't fit, you can't attach to the active site. So enzymes don't affect every molecule they see. They only affect ones that fit that shape. When the enzyme substrate complex is made, bonds in the substrate are weakened. Now notice I'm making and breaking bonds. I'm only illustrating breaking bonds. Like I said, it's kind of an easier picture to draw. And it makes it easier for my products to happen. So this is catabolism happening. I'm taking one large substrate and breaking it apart. The sheet that I gave you has two different examples on it. It has an example of anabolism and an example of catabolism. Does anyone have any questions they want to ask here? What is an enzyme doing and how is it doing it is totally a question that would be a long answer question on a quest, like the one that we're going to do on Friday. It will cover lessons 1 to 5. Lesson 5 is actually just a little lab activity that we're doing tomorrow about enzymes, so really today's notes are the end. But I'm telling you, what is an enzyme doing and how is it doing it is for sure something I will ask you to write about. Because there's lots of things that you can say. I want to hear in your own words what it's doing. Anyone want to ask anything here? So then the last thing that we need to talk about is how can I affect whether an enzyme is functioning at a high rate or a low rate or perhaps not at all. So we're going to talk about factors affecting enzyme activity. One of the factors is temperature. We said before, if temperature is too high or low, we can cause denaturation or coagulation possibly. And since an enzyme is a protein, if the protein is denatured, well, the enzyme can't do its function. Now I put a little graph here, and there's a very similar looking graph on page 215 of your textbook. And I just want to highlight 37. That is how warm your body is supposed to be, and most of the enzymes that we'll discuss are ones that exist in you. So for most enzymes, there is an optimal temperature. So notice how the curve of this graph kind of goes up as the temperature gets warmer. That's because if it's too cold, remember your proteins are inflexible, they don't function properly either. But as soon as you get too hot, their activity drops off. And that's where you get denaturation or maybe even permanent coagulation. So I'm not saying every enzyme has 37 as its optimum. But for us, 37 is the optimal temperature for a large majority of the enzymes that are functioning in our body. Now the second thing on my list is concentration, and I put 2 points here. Concentration of the substrate, concentration of the enzyme. One of them has a pretty big effect on how fast the reaction happens. One of them has a pretty minimal effect. The one that has the bigger effect is the concentration of the substrate. In general, more substrate equals a faster reaction. Now there's a limit to this. There's only so fast reactions can happen. But usually if I have more stuff to react, I will have more reaction. When I talk about my enzyme, the effect is much smaller. Why? Enzymes are reused. So I don't actually have to have a lot of them to have lots of reaction happening. So a lot of the time, changing the amount of enzyme will have very little effect. I'm not saying it has no effect at all, because there's a point where I'm going between 0 enzyme and some enzyme. That would make a difference. But once I have some enzyme present, increasing it quite a bit usually doesn't have a huge effect. So there's a smaller effect. It's because enzymes can be reused. But increasing the concentration of the substrate, generally, if there's more of it, my reaction will happen faster. Now pH is another, and the picture that I have here is very similar again to page 215 of your textbook. There's 2 little graphs there. One is of temperature, the other one is of pH. And it's to illustrate to you that some enzymes like it when the pH is low. Some enzymes really like it when the pH is high. So we'll say that this middle point is pH 7, and then there's my pH line there. So on this little curve, what I'm trying to illustrate is that there's not one number that's the best. This protein, so this enzyme, for example, has an optimum of around 2. It functions best when the pH is really low. And as the pH increases, even as the pH gets to 7, which is neutral, it does not function very well. This would be, for example, something that's in your stomach. It's acidic in your stomach. For an enzyme to work in your stomach, It has to function really well when the pH is really low. This enzyme over here has its optimum around 9. This is something more typical of your lower digestive tract, so not your stomach, your intestines somewhere. The pH is a bit higher there, enzymes function better. So not every enzyme has the same optimum pH. I would not expect you to memorize this at this point in time, but I would expect you to be able to read a graph. And if I showed you a graph of a pH versus enzyme function, I would expect you to be able to tell me what's that enzyme's optimum pH. So this type of question here is more about interpreting data than it is about memorizing a particular number. Does anyone have questions here that they'd like to ask? Last one, second last one maybe. There are other things that affect enzyme activity. Those were all physical things, conditions that I could change. These factors are other substances. So on the left, I have cofactors and coenzymes. I put them together because they're usually both positive. They would help an enzyme do its job, make the reaction happen faster. On the right, I put inhibitors and negative feedback because they generally reduce the function of the enzyme, make the reaction happen slower. Now on the cofactor and coenzyme side, cofactors tend to be minerals. Coenzymes tend to be vitamins. Now you might have heard it's important for you to take vitamins and minerals. Does anyone know the differences between a vitamin and a mineral? Usually people don't, and it's funny because you probably are aware that you need them. Oh, did you have your hand up? You can say. Vitamins are often found in fruits and vegetables. You're not wrong there. Minerals can be found there too. So where they're found isn't the thing that usually differentiates them, because they could both be found in a fruit or a vegetable. It's actually what they are. A mineral is inorganic. Vitamins are organic. Anybody remember what that means? Not the pesticide thing that they talk about at the grocery store, but chemically. What does something have to have if it's an organic compound? Yeah, carbon, that's right. So vitamins have carbon in them, minerals don't. Minerals are usually individual elements like iron, magnesium, zinc. Vitamins are more complex molecules that have carbon, usually oxygen, hydrogen, other things like that, but they have lots of atoms put together. So we have cofactors and coenzymes. What's the difference? Well, they're both helping, so they're both making enzyme activity higher. Cofactors attach somewhere, and you're going to say that's super vague. What kind of description is that? Coenzymes attach to the active site. And I'll add the word else. Cofactors attach somewhere else, not to the active site. Coenzymes are part of the active site. They help it become the correct shape. They finish it. Both of them complete the shape, let's say. So in both cases, imagine that there's like a space, a hole in the enzyme, and the cofactor or the coenzyme is filling it. For the cofactor, it's not attaching on the active site. So in my picture, there's like literally a hole right there. And the metal, which is a mineral, just fills that spot. On my coenzyme picture, This green part here is the coenzyme, and it's like finishing the shape of the active site, making it so that it fits properly to the substrate. But if you wanted to sum up what both of them are doing, they're completing the shape. They're finishing it off. Now on the other side, we have our negative things. And not all negative things are bad, hey? All we're saying is that they're reducing enzyme activity, but honestly, sometimes that's what we want. There are a couple of types of inhibitors. There is something called a competitive inhibitor, and there's something called a non-competitive inhibitor. I'll just move this over here. So a competitive inhibitor is something that literally blocks the active site. So we use the word competitive because there is competition for the active site. It's something that will also fit perfectly in the active site, and so it'll prevent the substrate from going there. So you'll see I've got a little picture there of that happening. In the first part of the picture, my substrate, which is this yellow thing, can get into the enzyme and the reaction happens and it's broken apart. But in the second half, there's an inhibitor. It goes in, it blocks that spot, and now my poor little yellow substrate cannot get in. Now, like I said, inhibitors aren't always bad. Sometimes we don't want the reaction to happen anymore. I gave you two examples here of real things that happen. Sulfa drugs. You might be aware that about 40-ish years ago, it was discovered that there was a medication that women were taking while they were pregnant to reduce morning sickness, so to reduce nausea and queasiness and stuff, that was actually causing birth defects. This is the scenario I'm discussing here. A sulfa drug is something in that category, stuff that gets rid of nausea, heartburn, indigestion, that category. They inhibit folic acid, which is totally crucial to the development of a fetus. So that's an example of a bad competitive inhibitor. A good competitive inhibitor could be one of these. ACH is an abbreviation for a neurotransmitter molecule. It's the one that goes between your nerves and your muscles, telling it to contract. And so ACHase is the enzyme that breaks it down. Sometimes that's bad. If I break that down, it could be acting like a venom and making it so a person can't contract their muscles and they die. But there are people who have conditions where their muscles contract too much. They have tremors. The one I wrote is called myasthenia gravis, but someone who has Parkinson's, let's say, has the same sort of thing happening where they have too many muscle contractions. Sometimes I don't want my muscles to contract. And so in that sense, it could be used to treat conditions. Now, are all these specific examples ones I'm going to test you on? No. You could use them if you want to describe what's happening. What I really wanted to show you was that not all inhibitors are bad. Sometimes they're good. The other reason that competitive inhibitors can be good, and this is my favorite word in all of biology, if you asked anyone who took biology with me last year, they will tell you that I refer to homeostasis as my biology husband. I love this. This is my topic. I will ask you a million questions about it. You will do a project about it. It will be on your final exam. This word, homeostasis. Anyone know what homeostasis means? So I was watching The Big Bang Theory the other night, and there was a little part, and Sheldon says homeostasis to Penny, and he says, "Do you know what that means?" And she says, "Of course not. Why would I know what that means?" And I said, well, let's break down the word. Homeostasis. It's a state where everything is the same. That's what it means. So, homeostasis, constant state. Your body does not like change. Your body has a particular set of conditions just like any living organism, and it's trying to maintain everything in this tiny, narrow little range. Homeostasis. If something changes, your body will immediately react to try to fix it. So if you get cold, you shiver to warm up. If you get hot, you sweat to cool off. Your body is always trying to react to fix this. So, how does this involve enzymes? There's something called negative feedback. Negative feedback is when a substance inhibits its own reaction. So let's say, for example, that my body's making insulin, and then we realize that there's enough insulin. The molecule of insulin goes back and stops the reaction from happening. It's called negative feedback, and we usually use the term loop. Very often, a substance that your body is making will inhibit its own reaction once enough of it has been produced. And that's how your body maintains homeostasis, in part, because it recognizes, oh, I was making testosterone, now I have enough, stop. Stop making testosterone. Now the other way is non-competitive inhibition. It's a little harder to illustrate that because it's simply when a molecule is not blocking the active site. Usually it's doing like what a cofactor is doing. It's attaching somewhere else and affecting the shape of the enzyme, but the exact spot is not specific. A heavy metal functions that way.